Well, welcome, and thank you for taking the time to be here, and especially all the international people, which is just wonderful. I promise that I'm going to come to the US at some point and meet all you guys, but it'll have to wait for another year. But anyway, today I'm talking about octocorals. Partly because I know most of you are aware that I've just written a guide to the octocorals, but also because in doing that, I was really turned on to them in a way that I just hadn't been for all the years that I've been diving. And my background, I'm a believe it or not, a paleontologist, a fossil person, and I started out being interested in fossil corals and fossil reefs, and that kind of went sideways into how to identify corals and looking at modern reefs to help understand the fossil ones that we were looking at in New Guinea, which is where I worked. And then I sort of kind of left research science and became more of a science communicator, and long story short, my sort of life passion has been to pursue the communication of knowledge and to create tools which are user friendly and to unlock the science that is already lying around in many cases and make it more accessible. So that's kind of what drives me, and I'm just going to. I've been asked to show you some photos of me. The good-looking person on the left is my wife, Rachel. She's got an Octocoral Finder in her hot little hand, and we've both been very busy this year. I was very busy producing this book, and she was very busy being a scientist working in reef restoration. And we went on a 6-week dive holiday to Indonesia, and I wouldn't call it a holiday really. It was 6 weeks taking photos for the next edition of the Coral Finder and the Octocoral Finder. And so we took 29,000 photos in 6 weeks. And but Rachel decided to teach herself the octocorals. And in the time that we were messing around, she recognized 50 different genera using this tool, actually more than 50. And the point is that I think a lot of people just are not really tuned in to what's there. And the octocorals are just as diverse as the hard corals, but they just don't get much of a Guernsey. And so today we're going to look more closely at them to do that. This photo, when we were in Bali, part of this last trip, I bumped into a whole bunch of people who we trained in coral identification in 2012. And if you wanted to ask what gets me really excited about life, the universe, and everything, is getting other people excited about life, the universe, and everything. And in this case, these guys work in the dive industry in Bali. They did my Coral ID course over a decade ago, well over a decade ago, I think. And they are still in the industry, but they are respected, knowledgeable people communicating information to their peers and to the public. And anyone who's worked in Indonesia and met the guides there, like they're just legendary for being able to find things. But also, it was really satisfying to meet a bunch of people who had stayed on and built careers. And I think building capacity through good knowledge and good tools for communicating science is a really important way forward to enriching everyone's appreciation. And in this case, these people are, every day are in the industry communicating to the people who are experiencing the reefs around Bali. This is just the last photo before we get on with the talk, but this is at a place called Papua Explorers in Raja Ampat. And every year I run a program called Coral Explorers, which is an introduction not just to corals but to everything you might see on a reef, but with more of a focus on the invertebrate groups rather than the kind of fishes and turtles and big things. And it also has a framework where we start with an explanation of how Raja Ampat formed, and so we look at the plate tectonics, and then we move on through each of the main groups of organisms that you might find on any given dive, and we just look at practical ways for recognizing them and how to separate the different groups. And so there's no Latin involved, but there's a lot of just stuff you can see with your eyes, and this is really key to getting people excited about diving and about reefs. And it's the most satisfying thing I do every year is seeing the lights come on with a bunch of divers who have traveled halfway around the world to see these amazing destinations, but also to feel like they're really learning and enriching themselves. And this is why capacity building, ocean literacy is so important. Okay, so well may we say, I should do this in Gough Whitlam's voice. Now, to our American people, this today in Australia is a very famous day. It's 50 years since an unelected representative of Her Majesty the Queen sacked the Prime Minister of Australia. So you guys will probably have no idea what I'm talking about, but this 50-year anniversary is all over the news. And this is the Prime Minister, and he said something like, well, may we say God save the Queen, because nothing will save the Governor-General. That I'm saying to you today, well, may we say Octocorals are amazing, because everybody knows that fish are stinky. And I have to sort of find a way to cut through to get people to, just acknowledge that this stuff is here. The things that you see on the seafloor, for many people, they've just become wallpaper, and they are richly endowed with behaviors and fascinating things, and that's what I'm really here for. So maintain your rage and enthusiasm, comrade, because we're going to be going on a trip through the wonderful world of octocorals. So in this talk, we're just going to look very briefly at the difference between hexacorals and octocorals, and that's basically the difference between hard corals and soft corals, sea fans, sea whips. Then we're going to just introduce those four groups, because that's what the Octocoral Finder is dealing with. It's helping people to identify and recognize consistently these main groups. And then we're going to look at what these things do physically and the ecological relationships that they have with the reef and around the reef. And we're even going to look at how some of these things actually contribute to reef structure. There are actually reef building octocorals, which I think is an important thing for people to be aware of. And then right at the very end, and I promise not to make too much of an ad of it, but we'll just do a little quick intro to the Octocoral Finder itself. And there'll be a discount offered if anyone's interested in a copy, a 20% discount at the end of the show. Okay, to why we're all here, the polyp clan. And, there's it's very easy to be overwhelmed on a reef. There's just this blizzard of biodiversity. But what we really have is two major groups that are visually prominent, and they are the hard corals and the soft corals. And most people don't kind of refer to things as hexacorals and octocorals, but I am going to do that just so that we can sort of talk collectively about all the things that are in both these groups and how to separate them. So hard corals, hexacorals, are known as for having a six-fold symmetry. So you have a skeleton that is formed by a veneer of tissue which is only 2 cell layers thick. So where you can see, can someone just let me know, can you see my mouse wiggling on the screen here? Yeah, okay, thanks. So you can see that little drape of tissue. And when you look at any of the amazing architecture that comes from in the hard coral world, all of that is secreted by the lowermost of those 2 cell layers. So in a sense, hard corals actually are extruding, or they almost, you could say they have an external skeleton, because it's only the veneer of tissue on the outside that is alive, and it's only 2 cell layers thick. And everything is created by the lower side of that veneer, and that is kind of external to the animal. It's a funny way to think about it, but it's sort of something to wrap your head around. So if you look at a brand new settled coral, like this one at the top left here, you'll actually note that there are 6 sort of structural elements. They're called septa. And so this is the only time you get to see the hex in a hexacoral. But it's just important to sort of kind of convince you that it's there, it's real. And so hexacorals also have tentacles in multiples of 6, but very, very quickly you can't see that, and so you can't count it, and you shouldn't even bother trying. It's just too hard. So hard corals, you are dismissed. That's all I wanted to point out, because what we're really here to do is look at the octocorals, and we're basically looking at 4 main groups: the sea fans, the sea whips, the sea pens, and the soft corals. And they are characterized in a way that you can always know that you're looking at an octocoral, because they have 8 feathery tentacles, and they're big enough that you can count them. So they're much more sort of user-friendly in that sense. But most of them do not have a solid skeleton, although there are some exceptions. But they have basically a soft sac of tissue in which there are tiny skeletal elements like little spines called sclerotes. Now, these things are, there's about 3,500 species of octocorals. Most of them are in the deep ocean. Now, this thing here is a deep sea pen, but it has 8 tentacles with these, when I say feathery, they're things called pinnules. They're the little sort of tentacly like things on the tentacles. But what's important to know is that there are 80 genera, approximately, shallow water genera, that are around reefs. And so if you do a lot of diving, you are likely to see around 80 genera of octocorals. Now, there'll be a mixture of whips, fans, pens, and softies. But what's kind of interesting is, oops, sorry, I need to come back. Just in terms of those 80 genera, the interesting thing is that's about the same number of genera as there are hard corals in the same equivalent environment. We're not talking about in the deep ocean or deeper waters, but basically you might be surprised to know that there's an equivalent diversity at the genus level, and probably almost at the species level, of both octocorals and hexacorals. Now, in terms of why don't we know much about these things? Why are they sort of like a fairly mysterious group that haven't really had much airtime, and they're not well understood as a group? It's probably in part because Whoops, we got someone on. Could you go to mute? Yeah. And part of the reason that is a lot of these things have been described from deep sea sampling. And so we're talking like dredges and trawls, and things get described, and they're described in a fairly unhelpful way. That is by looking down a microscope at the sclerites, and so the publications that describe the species and the genera are not very user friendly. But you can always tell when you're looking at an octocoral, because no matter what group it comes from, it'll have 8 feathery tentacles. So they're easy to identify as a group. Now, it's also important to note that probably in the shallow water sense, about half of them also have the same or similar symbiotic algae as to the hard corals. Now, the taxonomy of the octocorals is thorny. I was going to say thornier, but pun intended, because you look at this guy, Tony and someone else's favorite octocoral, Dendronepthia. You basically looking at something that, when up close, it looks like a spiky broccoli, and these things are full of little sclerites, and they're fascinating because they're there to provide a certain rigidity to the thing so that it can flow, be flexible. But when you look more closely at it, you can see that they also are doing other jobs. And you can see here, this very magnified image, you can see that these sclerites are supporting the polyps, and that inside the polyps themselves are more sclerites which are doing work of making the polyp functional. Now, all of this has to be flexible enough to allow this thing to expand and contract, because that's what these guys do. They're all about expanding and contracting. But it's interesting that they are made up with a whole raft of different sclerites of different sizes doing different jobs. And the actual taxonomy, the scientific descriptions, are based on collecting sclerites from different parts of the colony. So if you just look in here, you can see different sizes, different colours. Not that the colours are particularly important often, but as you go round the colony, maybe down the stem towards the base, a taxonomist will sample from different places on the branches and then sample the polyps themselves, and they dissolve the sclerites out. And when they are doing a species description, they end up with a portfolio of sclerites as part of their description, and they will have come from different parts of the colony. Now, the sclerites in the little black and white there, they're not the sclerites from this particular coral, but you can see there's an example there of 2 different kinds of sclerites. But it's quite possible to have many different kinds of sclerites in a species description for a soft coral or an octocoral. So, sea pens. We have the song remains the same. Very different looking things. They have a bulb buried in the sediment, and these horizontal feeding structures which are lined with polyps. And sometimes they're almost cylindrical and sausage-like. But basically the song remains the same. There are 8 feathery tentacles on the polyps, and so it's instantly recognizable as an octocoral. And yes, there is a rigid axis running down this central structure here. Fans, whips, and gorgonians, they all kind of belong together in a group we're talking about true sea fans and true gorgonians and true sea whips, in that they all have an axis, and the axis is made of a horny substance called or a scleroprotein called gorgonin. And so if you see a dead sea fan, usually it's going to be black, like this one in the center here, because the gorgonin is this black axis. And it provides flexibility to the colony. So what gorgonin does for these gigantic colonies of sea fans and things is it provides them with a flexible way to present many thousands and thousands of polyps to the current. So they effectively become gigantic two-way feeding sieves. And when they're busy being octocorals, they're busy feeding. And but the current will stop, and then it will go the other way, and these things will strain the waters when the current changes direction. But anyone who is kind of dived on a steep reef walls or ridges where there are fast-flowing currents, you know why the gorgonians are where they are. It's basically telling you that you're probably going to get washed away if you hang around here too much longer. Now, there's quite a lot of variability in these groups, and so we're not going to get into the taxonomy at all, but I'm just making the point that among the things that kind of look like sea fans, you'll find that there are other groups, and mostly the way that you recognize that is they have different arrangements of something that is like an axis that allows them to be flexible and present a feeding colony to the currents. And so in the bamboo corals, you have alternating calcite and gorgonian segments, and the gorgonin segments are, again, that's what's flexible. And there's just lots of these things, and particularly in the deeper ocean, there's many species in these other groups. But you might have also heard of things like precious corals. They have yet a different style of axis. They have a sort of a congealed sclerites. Anyway, the point I'm just making is that it's not quite as simple as I presented it to you. There's always traps for young players, and here's a good one. This is a thing that looks like a sea fan. They can be very large, well over 1 meter across. And when you look on the reverse side, what you'll see is it doesn't have a rod. It's not an axis. It's actually, this is a soft coral that is pretending to be a sea fan. And so it's managed to, in the sort of the orange branching rod, sort of the thicker branches, it actually does congeal all of its sclerites together to make something that is the equivalent of a gorgonian axis. But when you get to the branch ends, you see that this thing's actually sort of like an encrusting soft coral, and it's kind of wrapping itself around to pretend it's a gorgonian. So always have a look at both sides of your sea fans. So, in general, what else would you say about octocorals? They are probably the most significant thing, particularly in the soft corals and sea pens, is that they have this kind of hydraulic use of water to get things done. And so you can see here, this guy on the left, Scleronepthia, is up and about and feeding. And as soon as that current stops, that all stops, and the thing will just go, I can't be bothered. And that's what it looks like when it's not feeding. Likewise, everyone's favourite coral, Dendronepthia, when it's up and about and feeding, it pumps itself through water, full of water, mainly through the polyps, but also through some other structures, and it inflates itself. And as soon as conditions are not conducive to feeding, it will just pump that water out and turn itself into a very spiky little broccoli thing. And that's what's really intriguing about these guys, is they do not waste energy being big when they don't have to. If they're not getting something out of it, they're out of there. So this is kind of what causes a lot of problems for people trying to just kind of get into octocorals, and is that with hard corals, there's a lot of them, and they change form with environment, but that skeleton is kind of always the same. You come back an hour later or a week later or a month later, it looks more or less the same. But if you come back and look at an octocoral an hour later, particularly if it's feeding or not feeding, and particularly the soft corals and sea pens, they often look entirely different. And so this is part of the problem of getting that basic pattern recognition going that people use to kind of teach themselves. We're good at, humans are good at pattern recognition, but unless you actually see all of the bits, between fully inflated and fully contracted, it's hard to know what the actual animal can do. And I think our aquarium friends here have us at a disadvantage. They can often see this stuff in their tanks, and they spend a lot of time working on flow to make their corals and octocorals happy, and that's how you get to learn about what these things are really like and what they can actually do. So, but if you're diving, basically just have a good conscious look. Ask yourself, all right, is the one on the right the same as the one on the left? And usually, if you just look around the colony, you'll find a halfway version somewhere between the two. And so this is step one for teaching yourself octocoral genera, is have a good look. Now, this is a Dendronepthia style octocoral, and it's been, this is colony variation over one day. And you can see here that these things are expanding and contracting. Now, we're going to have an opportunity to watch this clip again in a second, but I'm just going to point out to you that you can see current velocity in red there. So just watch the number, and when it goes low, you can see that the corals respond more or less instantly to the variation in current speed. So hard corals have a skeleton with a veneer of 2 cell layers, and all of that amazing architecture is built by the lower surface of that second cell layer. And the soft corals, and to more or less the same way, all the octocorals basically live in a world where sclerites are king, and there are many different types of sclerites associated with different parts of the colony. And if you're a soft coral, you're more or less a sack of jelly with sclerites to give it a little bit of rigidity. Okay, so we're just going to look a little more closely at feeding. Hard corals or hexacorals and octocorals, basically the difference is that the hard corals in the main have simple tentacles. They don't have pinnules. They don't have these little structures on them. and they have larger stinging cells. And so by contrast with the octocorals, the pinnules lining the tentacles, and the stinging cells are smaller, and they've got less capacity to kind of apprehend prey. And so when you, I remember spending ages once when I was, a natural history filmmaker, and I was trying to get a soft coral to eat brine shrimp as part of a wall of mouths sequence that we were trying to film. And I just, like the brine shrimp would just hit these polyps on these soft corals, and they would just bounce off. Whereas if you did the same thing to a hard coral, there's a reaction. They get stung, then they get dragged in and eaten. And so they're very different beasties, and there's a reason for that. They're doing very different things. So octocorals don't compete directly with hard corals for the same food. So if on the right, that was the size of your little zooplankton, like a copepod, the equivalent food that the octocorals are eating are in a different classes. They're in the bacteria plankton and the microzooplankton and the ciliates and phytoplankton, as in plant plankton. And they're a whole order of magnitude smaller than the big buzzing copepods. And so for a diver, if you're on a night dive and you hold your torch up and then suddenly you've got thousands of things buzzing around in front of you, they're going to be the copepods. They're the things that the corals are feeding on, and you won't even see the other things that are more so the prey for octocorals. So what this means is that they don't compete for space, in the sense that they do compete for space, but they're not competing for food. And so they're doing different jobs in the same habitat. So a lot of octocorals, have pinnules that are developed to different degrees, and it seems like this, in some ways, is basically all about increasing surface area. And these stinging cells that are on the octocorals, they're more like fly paper. If you can imagine, like sticky paper, and the particles that are traveling through the pinnules, they've only got to touch it, and they'll stick. These microparticles will stick to the pinnules, and then they are engaged in a flow of mucus. And you can see that image, the video on the right. That is a microscopic imaging of the cilia that are in the mucus layer on more or less on any coral, any octocoral, any hexacoral. They have a mucus layer, and that mucus layer is full of beating hairs, beating cilia. And in the octocorals, the food particles, you generally can't see them, but they're always arriving and being shunted down those tentacles, down the pinnules, down the central groove of each of the tentacles and into the mouth. So that wall of mouths effect is actually very efficient at collecting energy. It's just that you can't see what's really going on. Now, this is another variation on having a lot of surface area, and not all octocorals do this, but why would you pulse? And it's, I think, it was a way of making water flow over that high surface area. I don't know that for a fact, but I just presume it. So, just some other basic, interesting sort of ecological sort of observations about octocorals. Yes, some of them do have hard skeletons, which again, these are made of sort of congealed sclerites. But what's interesting is that of all the corals to have coloured skeletons, this one and another one we're about to see, are octocorals. And so 8 feathery tentacles and a skeleton made of congealed sclerites, and big enough to actually form quite large structures, like that can be up to 1 metre across. And Tubipora, the organ pipe coral, is well known, but what's kind of less well known is that it forms a symbiosis occasionally with a sponge. And if you look inside the hole here on the lower right, you can actually see the red organ pipes. And on the outside of the sponge are the 8 feathery tentacles. And so when you look at the left, you're looking at the actual classical nodular sponge morphology, and the oscule, the big hole at the top, is the exhalant part of the sponge. And somehow, and it's still a mystery to me, but how these arrangements are arrived at over evolutionary time is still just remarkable. Who's controlling who here? We've got a sort of a weird arrangement whereby presumably the sponge is creating flow, and that flow has to pass through and over the pinnules, which is what the octocoral's getting out of it. And then the sponge matter is existing in and around the octocoral structure. So just how these arrangements come to be. If I did have a time machine and if I was God, I would this would be high on my list of things to work out how it happened. Now, a little bit of news from the research front. We've been talking to, I've been looking at when will I do the next edition of the Coral Finder, for anyone who knows what that is. And I've just heard that the family Argyricidae, which is Pavona and Leptoceras and corals like that, has just been undergone a major revision, and the genus Pavona is being split into 7 genera, not species, 7 genera in Pavona. So this is all going to be published next year, and same thing in Leptoceras is going to be broken up into several things as well. And so I've completely abandoned any idea of publishing the new Coral Finder before that all of that science comes out. But what I also heard was that the same lab, and this is all coming out of Kaust in the Red Sea, Francesca Benzoni's lab, that someone has collected Tubipora from across the entire Indo Pacific, including East Africa and the Red Sea, right through to the West and Central Pacific. And I think that there's likely to be 6 new species of Tubipora, and we always kind of suspected that because if you just take photos of them, you can see that the tentacles and pinnules are quite diverse, even though they've all got the same red skeleton to look at simplistically. But expect that to come out next year, and frankly, I'll be quite glad because I got annoyed with having to sort of think these are all the same thing, which obviously they're not. Now, here's another skeleton-forming octocoral. So you've got 8 feathery tentacles there, and on the left-hand side you can see 2 species of the blue coral, Heliopora, and just to prove that it's got a blue skeleton, but usually it looks brown or cream or grey underwater. But it too is a skeleton-forming octocoral. Now, ecologically, you can just generalize about the octocorals. If they're brown or cream or muted colors, they probably have the plant cells, the photosymbionts. And in general, if you have a coral that is bright colors or saturated colors, it probably doesn't have the photosymbionts, and it's also likely to be kind of deeper and in overhangs and caves and things and on walls. But there are some groups that seem to be halfway house. Things like Stereonepthia seem to be, they have species that have saturated colors and species that are also cream and brown. So it's not like I don't think it's a completely hard and fast rule, but it's a good guide to what your octocoral is going to want to do in terms of nutrition. So in any scene, you can see here on the extreme left and the extreme right, we've got 2 brown soft corals, and in the middle a brown hard coral, and on the left-hand blue one is what most people would call a cave coral, and it's a hot pink thing, not a great photo, sorry, and a bright red soft coral there. And so these are non-photosynthetic, and these 3 with the red outlines are photosynthetic. So again, interesting. Not only do the octocorals compete for different food and a different size, but they also have different strategies for making a living as well. So we already saw this, but just to reiterate, why, if there's a no current, why spend money, energy, keeping the whole show up and about? And it's just interesting to see how responsive these creatures are to their feeding source. Okay, so reproduction. A lot of these things are broadcast spawners, and I don't really know all that much about this. You'd probably be better off speaking to someone else about it, but just from a basic natural history point of view, you would quite likely be able to go and see octocorals spawning, just like you can go and see hard coral spawning. And if you actually take the time to look closely, because they are transparent, you get an advantage that you don't have with hard corals. With hard corals, you can't see inside the skeleton generally, but in the days and weeks, or the days and hours, I suppose, leading up to spawning, you can see this female here. You can actually see the eggs lining gastrodermal canals, I suppose they are, gastrovascular canals. And so this one is a female, and if you just leave that image seared to your retina, you can see here this is a male with a kind of a paler and less globular looking gametes. Now, like everything in biology, it's more complicated than that. It isn't just a question of males and females or hermaphrodites. There's all sorts of asexual shenanigans going on. And you can see in this bucket here, this coral has been in a bucket overnight, and it shed little polyps. And so this thing on the top left here is probably in close-up on the right, and you can see these little root-like processes, they call them, but basically they are holdfasts. And these corals are shedding babies all the time. And as if you know what happens in hard corals, you know that there's brooding in hard corals, and things like Stylophora and Poeciloptera are famous for brooding. So they do reproduce sexually on 1 or 2 moons each year, but they are constantly trying to brood and pop out little internally fertilized, fully built larvae as well. So again, next time if you're a diver or if you're an aquarist, have a look around the base of your colonies, and you should probably find these guys floating about. Now, something that I'd never heard of before until I started looking at octocorals was the blue coral. Not only is it got a coloured skeleton, which is pretty remarkable in itself. The 2 brightly coloured skeletons in the whole coral world are soft corals, Tubipora and Heliopora, having red and blue skeletons. But the Heliopora also brood larvae, which they fertilize internally, brood the larvae up to a point, and then extrude it, release it, and the larvae then attach themselves around the base of the stalk of the polyps. And that's where they remain while they're developing outside of the actual polyp. So this is a fascinating little bit of biology. And if you see, what I suspect is in the field you quite often see Heliopora bright white. Like most of the time it's kind of blue, like the top left-hand corner, or blue-brown. But every now and then you see one and it's frosted with polyps out during the day. And it seems a bit unusual. And what I suspect is when they are in the brooding phase, that those tentacles are up and out in order to provide the cover that these larvae are having or need. And so next time you see a frosty white Heliopora in your tank or in the field, go and have a good look and see what you can see. Now, similarly, there's a lot of asexual dropping of tips and dropping of little bits. Sometimes you see soft corals that just look like they're melting. And in this case we're looking at a sea whip, which is one of these guys that actually has a gorgonin axis. So this is interesting, but whenever you see Junesiella, as you don't see one, you see many of them in a cluster. They are clumpy, and the reason they're clumpy is they're always dropping tips, and so that's why you have forests of sea whips. So just another thing to look at when you're diving. You'll see the decaying. They, like these are about 5 to 10 centimeters, and they decay and then fall to the ground and roll around and eventually turn themselves back into another member of the forest. So lots of runners and this kind of melting, drooping thing, which I'm sorry I don't have a good photo of, but I think I got one on this last trip. I just haven't found it yet because it takes a while to log 29,000 photographs. But asexual runners are another strategy for these guys. So, okay, octocorals as reef builders. So we met briefly Tubipora, the organ pipe coral, Heliopora, the blue coral. But another important critter is Sclerophytum. Now, all of you guys, I'm sure, just went, what? That's Sinularia, but that's why you need an Octocoral Finder, because the Octocoral Finder is across all of the recent changes due to molecular taxonomy. And so the things that are now called Sinularia, there's just 2 species, and they are little brown ear-like things, like flaps, Sinularia brassica, and some other one I've forgotten. But all the things that have big fleshy bases and digit-like lobes, branching lobes, are now called Sclerophytum. Now, what's interesting about Sclerophytum is that just some of these species are prone to taking the sclerites that we've been looked at in these other ones, you've seen them, and they concentrate them through the base of their skeleton, base of the colony, and turn it into a reef rock. So the thing in the foreground is actually where some soft corals had been living. They're not there anymore. But these, you can see here, this is a spiculite. This is the technical name. But these things can be as thick as the trunk of your thigh. They're really substantial. And if you were to smash it open with a hammer, that's what you'd see inside, is the spicules that live in the octocoral are deposited into a thing that allows it, a structure that allows it to get off the bottom, move towards the light, and kind of also get a bit more current. So you can see here, they can be quite large. Someone just sent me this video last week. It's in Hawaii, of all places. So octocorals are, in fact, significant reef builders in a way that most divers are unaware of. And I don't think that they, are in any way going to outcompete coralline algae and hard corals as in terms of reef bulk, but it's important to give them their due. And next time you're walking along a beach, and if it's a beach that's at the back of a shallow reef flat, like for those who live in this part of the world, Orpheus Island is a good place to see this sort of thing. The reef flat is often got quite a lot of spiculite on it, and this one is a storm-driven chunk of spiculite that was, probably weighs 10 kilograms. It's quite large. And just wrapping up now, but if you spend a lot of time, way too much time, taking photographs of octocorals for taxonomic purposes, to do an octocoral finder, for example, suddenly you start tripping over things, and there's just this whole amazing world of things that live on octocorals, and they're just incredible ecosystems in their own right, and with a long evolutionary history, as that Macaulay sponge example showed. And I just encourage everyone to have another better look, a closer look, anytime you can. So, in summary, there's a lot of these things, and most of them, the majority of them, are below 50 meters, but there are 80 genera, roughly, in the reef-associated shallow, less than 30-meter habitats. The diversity is essentially, at genus level, is the same as hard corals. They all really easy to recognize because they all have 8 feathery tentacles, and you don't need to know any Latin to say that's a sea fan, and that's a sea pen, and that's a sea whip, and that's a soft coral. You can be sure just by looking at the 8 tentacles. And if you want to learn more, I just encourage you to take photographs and make an effort, get a bit better at it, and suddenly you'll be able to do a lot more than you thought you could do with a simple camera like a TG7, which has good macro. You can actually do quite a lot of what has previously been thought was only something that a taxonomist could do. I encourage you to take a natural history interest in the octocorals, because you can do quite a lot with just a good photograph. Some of them are reef builders, though not many, but they are important contributors to the reef structure, and about 50% of them have some kind of, in the shallow water sense, some sort of relationship with photosymbionts. And they compete with hard corals for space, but not for food. So here's the ad at the end. My wife and I, Rachel, we've been committed to trying to bring the corals and now the octocorals into more of the interested person's realm. When we first started messing around with hard corals, people going, that's crazy, Corals of the World is 3 volumes, it's thousands of pages, how can you create a little 50 and now 80-page book that reflects that? Well, basically, you can, but we're not designing tools for species ID. What we're designing tools is to allow people to recognize things to genus. And we create visual front ends. So what I design are things called visual decision tools. And what they allow you to do is get to a point where you can see a collection of these things on the same page, and then your brain does its pattern search, pattern recognition thing. And so I've now done that as an introduction to the octocorals, and you can teach yourself 80 octocoral genera with not much effort, other than perhaps if you like to be perfectly honest, you'll get half of them just by looking, and the other half you should probably take photos if you want to be sure. We've got 5 editions of the Reef Finder, the Coral Finder, and this new Octocoral Finder is available from BYOGUIDES. Just to kind of give you a sense of where we've come from, what we've going to, you can see here there's 5 editions, and you can see the prototypes there that we were using in the early days. Do they work? Yes, they do. So the before and after workshop there. That's for 500 people, but the results are the same for 2,000 that we've now trained. But basically, if you can turn a problem into a visual comparison problem, then the human brain is, provided you don't overwhelm it, is capable of really resolving things very quickly. What is the enemy is big technical language and convoluted text. So basically the Coral Finder and the Octocoral Finder throw out most of the text, and they throws out most of the terminology, and it creates simple visual comparison pages which allow you, your supercomputer, your visual supercomputer that you carry around with you all the time, it allows it to do what it does best, which is choose between things that look about the same. Now, what with the Coral Finder, we've been able to build capacity really effectively in non-specialists, people who are interested. We're not talking about, everything being just for, scientists, research scientists, or PhD and postdocs. This is not about that. Although what I know is that next week I'm doing a training workshop at James Cook University, and it'll be just full of PhD and postdocs, because it's much easier to learn and problem solve if you've got a tool like this. And you can see here it's hard to sort of prove that this has had any real impact on science, but one way was you can see looking at the amount of publications that are using genus level data since the release of the Coral Finder. There's been a significant uptake, and I think that correlates with the fact that it works. With the Octocorals, basically most surveys, people will go, I know 3 Octocorals, I know Xenia, I know Sinularia, and I know Lobophytum. And I would say that in most cases people don't actually know Xenia, Lobophytum, and Sinularia. You think you do, but I'm sorry, it's not quite that simple anymore. Most surveys use schema like this, where things are reduced to morphotypes, and this tool allows you to move beyond that and to actually collect genus level data, or a combination of genus level data and morphotypes, because to be honest, quite a few of the Gorgonians are actually still quite tricky. But in any case, this tool is for you if you are a natural historian and you're interested. This tool rewards the self-directed learning. So you pick a shape off the front cover. That sends you to the visual index, which is 20 pages at the front of the book, which are compare and contrast of similar genera that match the shape and texture solution that you chose from the front page. And then you pick one of those and it gets sent. You then go to a gallery where you'll see much larger images and detailed photography and explanations. And so here you can see everyone's favorite coral, big fleshy thing, brightly colored. So you go branching, stalked, colored, and go to visual index pages 2 and 3. So you're in the visual index now, and all the things that are thick and fleshy and brightly colored are all on this page. You can see there's 1, 2, 3, 4, 5, 6, 7, 8 genera. And there's a terse description there, and I won't make you look at this in any way, but the point is that you now have very quickly put the best candidates on the table. You pick the ones you like, maybe you pick 2 or 3, and you go and have a look at them in their detailed genus galleries, and you can see this is just part of a 4-page gallery for Dendronepthyia. But the point is it works, and you can teach yourself, and there's very little Latin involved. So I think that's just about us. I just, if anyone's interested, I'm happy to provide Octocorals, the Octocoral Finder at a 20% discount at the moment. So you can just email me on that address if anyone is intrigued. And also, if I'm supplying them in Australia, they'll be on a slightly heavier grade of paper than the ones from the print-on-demand service that you get off the website. That is pretty well us. I hope that was of interest, and I'm going to pass you back to Jacinta, our moderator.
Wow, thanks so much, Russell. Really, really great presentation. I also wanted to thank everyone for joining us today for this reef conversation. And we'll try and get the recording up on YouTube by the end of this week, hopefully. Our next webinar is on the 26th of November with Professor Greta Peckel and Professor Jan Strognall on the impacts of climate change on natural systems. So please stick around and hopefully you can see that presentation as well. And if you want to catch up on any of our past presentations, they're all on YouTube.